Hello everyone, welcome to today's session. Today we are going to see class 12 biology, chapter 4, principles of inheritance and variation. In that we are going to see genetic disorders. Yeah, just start with the topic. Imagine that a child inherits dimples from one parent, curly hair from another, and even breadfruit from the family. So these are examples of normal inheritance. But sometimes what happens along with this normal traits, a person may also inherit diseases caused by changes in genes or chromosomes. Such diseases are called genetic disorders. The idea that diseases can run in families has been known for centuries. But after Gregor John Mendel's principles of inheritance were discovered, scientists understood that these disorders follow specific inheritance patterns. So every character in our body is controlled by genes present on DNA. Where it is located in the chromosome. So normally DNA passes from parents to children without change. However, changes called mutations also occur sometimes, yeah. So these mutation may alter genes or chromosomes and lead to inherited disorders. So genetic disorders are broadly classified into two major groups. One is the Mendelian disorder, yeah, you can able to see here. So which is caused by mutation in a single gene, and chromosomal disorders is caused by abnormalities in chromosome number or structure. So today we are going to see this chapter how these disorders are inherited and how scientists trace them using family history and also how they affect the human body. Yeah, let's start with the pedigree analysis. Why can't we study inheritance in humans like Mendel did? Yeah, Mendel performed controlled crosses. What Mendel did? He performed controlled crosses using pea plants because plants reproduce quickly and scientists can choose which plants to cross. Is it possible in humans? See, in humans this is impossible because human generation time is very long and also families have fewer children and also controlled mating is neither possible or nor it is ethical, right? So scientists they needed another method in order to study this inheritance. Yes, there comes the pedigree. So what is this pedigree analysis? So the alternative study, yeah. So the alternative is to study the family history of a trait over several generations. So this method they called as the pedigree analysis. So pedigree is a family tree. What is a pedigree? Pedigree is a family tree showing how a particular trait like a disease or abnormality is inherited from one generation to another. So instead of performing crosses, scientists they observe grandparents, parents, children and also grandchildren to understand inheritance. And what is the importance of this pedigree analysis? See, it helps scientists to trace inherited diseases, right? And also determine whether a disorder is inherited or not. And also to predict the chances of disease appearing in future children. And also identify the carriers and understand whether a disorder is dominant or recessive. And also determine whether a gene is autosomal or sex-linked. So pedigree analysis is one of the most important tools in human genetics. Let's see the pedigree symbols. You can able to see the pedigree symbols here. Yes. So these symbols, the first one, the square, it represents the male, and the circle, it represents the female, and this filled one, all these are the affected individuals, and you can able to see the mating. Yeah, the single line is mating, and this is the double line is mating between the individuals, that is relatives, and also parents above one, children below. These are the parents and these are the children, right? And also parents with male child affected with a disease. So this box is filled, right? So it is a diseased one. And also 5 unaffected offsprings. Yes. So because everyone use the same symbols, this pedigree can be understood anywhere in the world. Exam tip for you. Remember the standard pedigree symbols, and these are frequently asked in your CBSE examinations. Let's start with the Mendelian disorders. So what are this Mendelian disorder? So Mendelian disorders are genetic diseases which are caused by mutation or alteration in a single gene. Don't forget alteration or mutation in a single gene. Only one gene is involved. These disorders, they follow the Mendel's law of inheritance. What they follow? They follow Mendel's law of inheritance. So their inheritance patterns can easily be traced using the pedigree analysis, right? So some common Mendelian disorders I have given here, like hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, and thalassemia, right? And also dominant and recessive disorders also. So Mendelian disorders, it can be a dominant or recessive. So only if a dominant means only one defective allele is enough to produce the disease. Dominant means only one defective allele is enough to produce the disease. Example is myotonic dystrophy. I'll explain this fully now. Yeah. So recessive. Recessive means disease appears only when both alleles are defective. Yeah. Example is sickle cell anemia. And this pedigree analysis also helps us to distinguish whether a trait is dominant or recessive. Yes, let's see this one. We already know the square is the male and the circle is the female. And here, this is the affected female, right? Yeah, let's see this autosomal dominant example, myotonic dystrophy. So what happens here? See, only one dominant allele is needed to express the trait. So the affected individuals usually have an affected parent. So affected individuals usually they have a affected parent, and the trait appears in every generation. Can you able to see the trait? Yeah, whichever is crossed, no, it appears in every generation. Both males and females are equally affected. So this is the male and this is the female affected ones. And an affected parent and a normal parent, they have a 50% chance of passing the disorder to each child. So in the pedigree, what happens? The disorder is present in every generation. You can able to see disorder is present in every generation, and also every affected person has at least one affected parent. So every affected person has a one affected parent. Can you able to see? Yeah. So next one, autosomal recessive inheritance. So autosomal recessive inheritance, example is sickle cell anemia. So what is the characteristics of this? See, a person may inherit two recessive alleles, that is to be affected, and parents are often they are often normal carriers, and the trait may skip a generation. It may skip a generations also. Both males and females are equally affected. Here also both male and female are equally affected. So when two carriers, so this is a carrier, right? This is a carrier. So when two carriers have children, they have 25 percentage affected, 50 percentage of carriers, and 25 percentage normal. So in the pedigree, two unaffected parents. These are two unaffected parents. Can you able to see? So here also these are two unaffected parents. Yes. So the disease skips the generations, and more than one sibling may be affected while the parents remain unaffected. So what is the main difference between this autosomal as well as this autosomal dominant and autosomal recessive? See, in autosomal dominant, one dominant allele causes the disorder. Here, two recessive alleles are needed. It appears, this one appears in every generation. You can able to see it appears in every generation, and this may skip a generation. Here, the affected person usually has an affected parent. Here, parents are usually unaffected carriers. Yes, unaffected carriers. So here example is myotonic dystrophy. Here example is sickle cell anemia. Yes, so dominant means it won't skip generations. Recessive can skip generations because carriers are unaffected. Let's move on to the next one. Next, we are going to see sex-linked Mendelian disorders. The first one is the color blindness, right? So, color blindness is an X-linked recessive disorder. When it occurs due to mutation in genes. It occurs due to the mutation in the genes, yeah, controlling the red or green cone cells of the retina, which controls the red or green cone cells of the retina. So affected individuals, they cannot able to distinguish between the red and green colors, right? So why males are affected more here? Anyone can say? Yeah, you can give your answers in the comment box, children. Or if you have any doubt, you can ask in the comment box also. I'll explain you. Yeah, feel free to ask, right? So the genes are responsible are located on the X chromosome. So gene responsible, they are where they are located. They are located on the X chromosome. So males have only one X chromosome, right? So male is XY, and females have two X chromosomes. And male needs only one defective gene to become colorblind, but a female, they need two defective genes, and it makes the disorder much rarer. And according to our NCERT book, like around 8% of males are colorblind, and only 0.4% of females are affected. Okay, and what about the inheritance pattern? See, if mother is a carrier, yeah, if mother is a carrier, every son has a 50% chance of being colorblind. If like every daughter has a 50% chance of becoming a carrier, right? And a daughter becomes colorblind only. A daughter becomes colorblind only if mother is a carrier and father is colorblind. So this combination is very uncommon. Yes. So next you can able to see here, right? So next we are going to see the hemophilia. So hemophilia is also another X-linked recessive disorder. So what happens in this disease? One of the proteins involved in blood clotting is absent or is defective. So what happens? Even a small cut, it can cause a prolonged or continuous bleeding, and because the blood fails to clot normally. Why it happens? Blood fails to clot normally. And the disease usually it passes from an affected, yes, it passes from an unaffected carrier mother. Unaffected, you have to, yes, see here, unaffected carrier mother to her sons. A female affected with hemophilia is extremely rare. Why? Because her mother may be at least a carrier, her father must be hemophilic. So this hemophilic males historically had reduced survival, and this combination is uncommon. I can give you one example. Famous example is Queen Victoria, who was a carrier. So the disease spread to many royal families in Europe through her descendants. And what is the exam tip? So male express X-linked recessive disorders more frequently because they possess only one X chromosome. Let's move on to the next one. Next, we are going to see autosomal recessive disorders. Example, I told you already is sickle cell anemia. So, sickle cell anemia is an autosomal recessive disorder, and it is controlled by two alleles. You can able to see HBA, and this HBA is the normal one, and HBS, this is the mutant. And the genotypes, yeah, let's see the genotypes also. If HBA is a normal, HBS is a carrier, that is the sickle cell trait. And also next one is HBS is a diseased one. So the carriers, they appear healthy, but they can transmit the mutant gene to the children. So each child has 50% of inheriting the mutant allele from a carrier parent. Let's see the molecular basis for this one. When we take this molecular basis, see one of the most important molecular biology example, this one, right? And also a single base substitution occurs here, or the change occurs here. So what is happening here? GAG is changed into GUG. What happened? GAG is changed into GUG. So this changes the sixth amino acid in the beta globulin chain. So what is there in the beta globulin chain in the sixth place? It is glutamic acid. This glutamic acid has been changed to valine, VAL, yes. So a change in only one DNA base changes, one amino acid, and also it alters the entire hemoglobin molecule, right? And what is the effect on RBC? See, yeah, under low oxygen concentration, this mutant hemoglobin molecules, they polymerize. Yes, so because of this, the RBC, it changes from their normal biconcave disc shape. You know what is the shape of an RBC, right? So from their biconcave disc shape into a long sickle-shaped cells. And these sickle-shaped cells, what it does, it carries oxygen less efficiently, and also they block the blood vessels and they break very easily, and also it causes anemia and pain. Yeah, next we are going to see phenylketonuria. So phenylketonuria is an autosomal recessive metabolic disorder. It affects the individuals. Yes, it affected individuals. They lack the enzyme that converts the phenylalanin to tyrosine. What they lack? They lack the enzyme that converts phenylalanine to tyrosine. So as a result, what happens? This phenylalanine, it accumulates in the body. Yes, it is converted into phenylpyruvic acid and other toxic substances. These accumulate in the brain, it leading to mental retardation. And also the kidneys cannot reabsorb these substances. So they are excreted in the efficiently. So they are excreted in the urine. Thus the disease is an inborn error of metabolism. Yes. Thalassemia. So thalassemia is also an autosomal recessive blood disorder. Can you able to see thalassemia? Yeah. So both parents are usually unaffected carriers. What they are? They are unaffected carriers. So unlike the sickle cell anemia, the problem is reduced synthesis of globin chains. Reduced synthesis of globin chains. So hence this fewer hemoglobin molecules are produced and it results in severe anemia. We have two types in this thalassemia. One is the alpha and next one is the beta. In alpha thalassemia, it is controlled by HB, yeah, HbA1 and HbA2 genes. It is located on the chromosome 16. It is located on chromosome 16 and 4 genes are present in total. How many genes? 4 genes are present in total. Mutation or deletion of one or more gene, it reduces the alpha globin production. And the more genes affected, the more severe the disease. So next is the beta thalassemia. It is controlled by HBB. This one HBB gene, and it is located on chromosome number 11. Yeah, it is caused by mutation in one or both the HBB gene, one or both the HBB gene, and it leads to reduced beta globin production. Let's see a quick difference between this thalassemia and sickle cell anemia. See, this comparison is very important for your board examination. Yes, and also students used to confuse these two disorders. So remember this distinction. Yes, so thalassemia is a quantitative. How much quantity it is produced? So thalassemia is a quantitative defect. So too little globin is produced. Too little. Yes. Sickle cell anemia is qualitative. That is, the abnormal globulin is produced. Qualitative defect, abnormal globulin is produced. Chromosomal disorders. What are chromosomal disorders? See, unlike Mendelian disorders, chromosomal disorders, they are also caused by absence of chromosomes, extra chromosomes, or abnormal arrangement of the chromosomes. The entire chromosome is affected rather than a single gene, right? So normal human chromosome number, you should understand first. So humans normally possess 46 chromosomes. How many pairs? 23 pairs. They include 22 pairs of autosomes and one pair of sex chromosomes. So any change in chromosome number can produce serious abnormalities. Yeah. Next of all, let's see first this aneuploidy. So sometimes chromatids, they fail to separate during the cell division. This is called a non-disjunction. What is called as non-disjunction. So as a result, what happens? Cells gain or lose chromosomes. So this condition is called as aneuploidy. Cells gain or lose chromosomes. This condition is called aneuploidy. So examples are trisomy, that is one extra chromosome, or monosomy, one chromosome missing. Next one is polyploidy. Sometimes the cytokinesis fails after telophase. What happens? Sometimes cytokinesis fails after telophase. So instead of gaining one chromosome, the entire chromosome set is duplicated. Instead of gaining one chromosome, the entire chromosome set is duplicated. So this condition we called as the polyploidy. And polyploidy, it is common in plants, but it is very rare in humans. Next, we are going to see the Down syndrome. Down syndrome results from trisomy of chromosome 21. So the individual has 47 chromosomes. So this disorder, it was first described by Langdon Down, 1866. Who discovered it? Langdon Down in 1866. What are the features? Short stature, small round head, and flat back. Yeah, flat back of head. You can able to see here, flat back of head. So the picture. So I'm just saying, just to look the picture, children. Broad flat face. Yeah, and also furrowed, large wrinkled tongue, and partially open mouth, and also broad palm. How the palm is broad palm, and single traverse palm crease, and many fingerprint loops, and congenital heart disease, and also delayed physical development will be seen, and also delayed psychomotor development will be seen, and also mental retardation, right? So let's move on to the next one. So next is sex chromosomal disorders. First one, we are going to see the Klinefelter syndrome. How it occurs? Because of an extra X chromosomes in male. Generally, how many chromosomes we have? We have 46 chromosomes, right? In 23 pairs, but here 47 chromosomes are there, right? So features, just see this picture, is male appearance, and also feminine breast development, that is gynecomastia, and also sterility. So the individual has masculine development with some feminine characteristics. Next is the Turner syndrome. The picture is here. So here one X chromosome is missing and the karyotype is a 45. See, one is missing. So instead of 46, it is 45 XO. Yes. And what are the features? Is female, short stature, and also rudimentary ovaries and sterility, and also poor development of the secondary sexual characters. See, Turner's syndrome, it results from monosomy of the X chromosome. Yes. Let's move on to the, yes, let's have a quick revision. So this chapter, what we saw now, it teaches that inherited disorders arise because of changes in genes or chromosomes, right? And also this pedigree analysis is the primary tool, used to trace inheritance in humans since controlled crosses are impossible. And also this Mendelian disorders result from mutation in a single gene and also include color blindness, Haemophilia, Sickle cell anemia, Phenylketonuria, Thalassemia, and cystic fibrosis. So these disorders may be autosomal or sex-linked and dominant or recessive. So in contrast, chromosomal disorders arise due to abnormalities in chromosome. How these arise? Due to abnormalities in chromosome number or structure, and often because of aneuploidy, that is trisomy or monosomy, or in plants, that is polyploidy. An important example you should remember, that is the Down syndrome, right? So Down syndrome, that is trisomy 21, and Klinefelter's syndrome 47, that is XXY, and also Turner's syndrome, that is 45XO. So understanding these inheritance patterns, molecular basis, chromosome changes, and characteristic feature, this is very important for your CBSE board examination. Don't skip the video. Watch the full video. We'll see you in the next video. Thank you.